Welcome. I'm Yvonne. I'm a product manager at Lexogen, and today it is my pleasure to walk you through some Lexogen solutions and some best practice tips for RNA sequencing experiment. Before we start, I would like to introduce briefly Lexogen. Lexogen is the RNA expert biotech company. We are a group of scientists with a passion for RNA, and we aim to serve the scientific community to favor research progress. Lexogen was founded in 2007 and is based in Vienna, Austria, and in New Hampshire in the US. We at Lexogen develop our kits and services to empower our customers with innovative solutions for RNA research and transcriptomics. Lexogen sells directly in both Europe and the US and several countries around the world. Further, we partner with 30 distributors worldwide to make our products and services accessible around the globe. Lexogen offers products, consultation and support, NGS services, and several B2B partnering opportunities. Our technical support team is one of the best in our industry. We offer upfront consultation, help with experimental setup, offer walkthroughs through our protocols or hands-on training, and all of that even before you start your experiment. Lexogen has a variety of resources and tools also set up for our researchers to learn about RNA-Seq and experimental planning. But of course, we also support you during your experiments or help you with the analysis and interpretation afterwards. Our product portfolio includes kits for single-cell sequencing, differential expression profiling, and whole transcriptome analysis, among many others. Lexogen offers kits covering the whole RNA-Seq workflow, from sampling, RNA preparation, library prep, to NGS data analysis, and also universal NGS controls. Our expert NGS services team provides you with best-in-class tailor-made solutions for your specific project needs. From upfront consultation to help you with your experimental planning and setup, over sample extraction, quality control, library prep, and sequencing, we offer also data analysis and will deliver your data in a convenient report that you can directly use to advance your research. Simply send us your samples at every step in the process and let our team take care of the rest for you. If you decide to perform your experiments yourself, we have a variety of resources to offer support along the way. And in this part of the talk, I would like to focus on the best practice for RNA-Seq experiments and help you with some useful tips. We asked young researchers what are the major challenges in setting up RNA-Seq experiments. And most of them answered that it is the setting up it is the new methods, the protocols or kits they need to handle, and any other kind of handling related topics. In reality, however, the major challenge lies in the experimental planning. As you can see from the top of the graph, 3 major areas are there for the planning phase: the experimental design, the sequencing design, and the quality control planning. And already here, you need to consider how you want to sequence your samples, which library prep you should use, and what you will need for your data analysis in the end. This all happens before you dive into the experiment itself or even pick up a pipette. Experimental planning is the most crucial step in the RNA-Seq workflow, and when done right, it will set you up for success. To help guide you through the process whenever you may need it, we provide a free learning resource on our RNA lexicon. This has a chapter also on experimental design and the background information behind each step. You can access RNA lexicon through the QR code displayed, and you can also download a free ebook. Should you have any questions before setting up an experiment, don't hesitate to contact our technical support team for upfront consultation and help with your specific experimental needs. The major questions any researcher should ask before diving into RNA-Seq are, what are my research question and my aim? What type of data do I need to answer my questions? Does my sample type impose any restrictions? And how can I control my experiment to draw valid conclusions? So while we delve on our research question, the major part here is really what aim do we want to come out of our RNA-Seq experiment? Once this is answered, based on our research question, we can decide if we need qualitative or quantitative data. So for example, do you require information about the expression of a given transcript or gene, about the expression under certain conditions, or about the transcript properties? So for example, where and how it is spliced, and if there are differences under different conditions. This brings us directly to the method you may want to use for your experiment. This, again, is based on your need and your sample, and you have various choices. If you require information on more than gene expression levels, and you want to investigate also other types of RNA, not just mRNA, you should be looking into whole transcriptome RNA sequencing. It allows to assemble transcripts, detects isoforms, and can also deliver information about expression levels, of course. However, if you're mainly interested in mRNA and you require gene expression analysis, 3' mRNA sequencing is ideal for your experiment. Here you can sequence one read per transcript, which facilitates the data analysis significantly and will also save you costs on the sequencing experiment. While whole transcriptome sequencing requires complex data analysis steps, as you need to assemble the reads along the transcripts correctly, 3' mRNA-Seq accurately quantifies expression by straightforward read counting, as here, one read is generated per transcript at the 3' end of the mRNA. Lexogen offers solution for both methods. Choral RNA-Seq is ideal for this differential expression analysis, especially if you're interested in transcripts. For the analysis of transcripts, including non-polyadenylated and long non-coding RNAs. For the detection of isoforms, transcript variants, fusion transcripts, alternative splicing events, and other such applications. And also for non-polyadenylated samples, for example, for bacterial species. Quanseq 3' mRNA-Seq is the right choice if you're mainly interested in differential expression analysis. However, Quanseq can also be used for polyacid identification and for RNA kinetics. So if you are interested in synthesis or turnover rates of global RNA molecules in your sample, you can use Quanseq in combination with SLAM-seq. Also, Quanseq can be used for targeted sequencing, for example, with customized panels. Whole transcriptome sequencing covers the complete transcript and provides a uniform coverage from the 3' end to the 5' end or 5' end to the 3' end, which you can see here in the green curve. In contrast, 3' mRNA-Seq focuses the reads on the 3' UTR and the ultimate exon, so these parts are efficiently covered in sequencing experiments. This also means that due to the focusing of the reads, you typically can sequence such libraries with less depth and get enough information to do gene expression profiling. Ultimately, 3' mRNA-Seq also requires less computational resources. It reduces the turnover time for your data analysis, and it provides accurate expression information through read counting. So if you are interested in expression profiling, especially if you have many samples, 3' mRNA-Seq is the right approach for you. Whole transcriptome sequencing, on the other hand, can also resolve isoforms. Here, the data analysis is more complex and requires more computational resources, but it also delivers a higher level of information. To resolve the underlying isoforms, as you can imagine, whole transcriptome sequencing experiments require more sequencing depth than the 3' sequencing methods. This is another aspect to consider when planning your experiment. How much depth will you actually need to answer your questions? While 3' mRNA-Seq relies on the availability of a poly A tail and is thereby limited to eukaryotic samples, whole transcriptome sequencing can also be used for any other sample or any organism that does not contain a poly A tail. So if you are mainly interested in transcript isoforms or alternative splicing, or are working with non-eukaryotic organisms, whole transcriptome sequencing is the right choice for you. But how can you control your RNA-Seq experiments? Controls are a crucial part of all experiments and also of RNA-Seq. And there are different types of controls that you should use and consider. First up are the replicates. But how many replicates do you actually need? If you ask any bioinformatician, they will likely tell you the more the better, as a higher number of replicates will give you more significant data. However, RNA-Seq is still far too costly to include all the replicates possible, and therefore a compromise is needed. Here, it is also beneficial to check the requirements for data analysis. Several tools require at least 2 or 3 replicates per conditions to be actually used. Again, another thing that you need to consider when planning your differential expression analysis experiment. Also, appropriate negative controls should be included. These are commonly samples that are either non-treated controls or that are wild types when we're analyzing mutants, as well as background controls when we're doing any kind of pull-down or enrichment experiment. But how can you make sure that the whole workflow is set up correctly, does not vary too much over time, and why is that even important? In 2016, Nature published a survey asking more than 1,500 researchers if they had any concerns for experimental reproducibility. And 90% of researchers answered that they were aware of either a significant or a slight crisis. For RNA-Seq experiments, the lack of controls and detailed documentation often results in different outcomes and interpretations, or data analysis not being reproducible when repeated by a different lab on a published dataset. Technical variation as well is naturally observed in virtually every experiment. So measuring the degree of variation introduced by changes to the complete RNA-Seq workflow, these are expected, is required to be able to compare current and previous data sets or to evaluate experiments between different laboratories or across sites. Although we routinely include several controls in standard molecular biology experiments, using them in RNA-Seq is still not common. And that is despite the fact that variation and often bias could be introduced during each step in an RNA-Seq workflow. Lexogen therefore offers SIRVs, unique spike-in RNA variant controls that can be used to validate RNA-Seq workflows. SIRVs are artificial controls that allow to validate the capacity of complete workflow, to detect isoforms, new ones and known ones, quantify them, set up and validate experimental pipelines, and parameters. SIRVs also allow better quality control of RNA-Seq datasets that are obtained from different labs or over time or between different runs. SIRV controls are added during or after the extraction, and they are processed together with the RNA-seq samples. Typically, only approximately 1% of total reads are dedicated to the SIRVs. And even this little fraction is sufficient to validate the RNA-seq workflow and assess the performance and the limits of the given setup. As mentioned, SIRVs are artificial sequences. Therefore, they are universal and can be used with any sample type or species, and also in any kind of workflow. SIRVs are polyadenulated, so your library prep can either start from poly-enriched RNA, from rRNA-depleted RNA, or you could use total RNA. But what are SIRVs? SIRVs mimic the natural complexity of transcriptomes. The transcriptome has complexity in concentration differences between lowly expressed and extremely highly expressed RNA classes. And the SIRVs are designed as a family of modules where each module mimics a specific aspect of the natural transcriptome. The SIRV isoform module, for instance, mimics transcription, splicing, and alternative transcripts, while the ERCC module addresses the concentration dynamics, and the long SIRVs mimic the length complexity of natural transcriptomes. The ERCC module contains 92 transcripts that follow a one gene, one exon, one transcript layout, so they are quite simple when it comes to the transcript itself. However, the ERCC module assesses the dynamic complexity of transcripts with concentrations between 1 to 1 billion molecules. Therefore, it enables the input and read depth dependent detection and quantification and straightforward input-output correlation. This module serves as a standardized assessment for the dynamic range, the dose response, lower limit of detection, and full change response. Input-output correlations can be measured by making use of the differentially concentrated ERCC spike-in controls. As the input concentration, as it is in the mix, is known, it is possible to control how well the RNA-seq experiment actually picks up transcripts of the different concentration classes. And this allows us to evaluate the sensitivity of the workflow and assess where we have the detection limit and the quantification limit. This can also help us to adapt the experimental parameters, for example, the RNA input amount or the sequencing depth, to the need of our researchers. On the right, such an input-output correlation is shown for a quant-seq experiment. The narrow distribution along the diagonal confirms that quant-seq is ideal for gene expression profiling experiments, as it accurately covers a wide dynamic range, as shown for the ERCCs. The long SIRV module mimics length complexity, while the transcript of the isoform module and the ERCC modules are relatively short and are on average just 1 kb, with lengths up to 2.5 kb. We know that the human transcriptome contains much, much longer transcripts. And this is just one example for the species of human. So the long SIRV module contains 15 transcripts in different length classes, from 4 kb, 6 kb, 8 kb, 10 kb, and 12 kb, with 3 transcripts each. Long SIRVs also have a 1 transcript per gene setup, and similarly to the ERCCs, they do not have isoforms, but they help us to identify if our workflow can cover the complete long transcript from its start to its endpoint. With the long SIRVs module, we can assess the length complexity of the transcriptome. The SIRV isoform module, and this is the most complex one, mimics the isoform complexity of the transcriptome. It has 7 loci, and across these 7 loci, 69 isoforms, including antisense transcripts, are distributed. They are derived from human model genes, but they also have unique sequences. So there's no significant similarity to any known species, which means that they can be used with any organism. And this module probes for alternative splicing, alternative transcription start and end sites, overlapping genes, antisense transcripts, and it allows you fast detection and quantification of isoforms in your RNA-Seq workflow. So now, why is that important? After all, we do have good annotations for some of the organisms that we're working with. This is a schematic representation of what we're actually dealing with in most of the cases. Higher organisms are characterized by complex transcriptomes. Usually, we have, per gene, multiple transcript isoforms. Sequencing reads, however, especially from short-read sequencing, are generated for each isoform, and then they need to be correctly attributed to the respective transcripts. So we have to sort them into the correct order in silico after the experiment. However, also the best genome annotations are not 100% correct. Also, there could be isoforms in the annotation that are derived from a bulk RNA-Seq experiment and that are not expressed in every sample, but only in a subtype of cells or tissues. So how can we then find out if our reads are assigned correctly and which isoforms are truly expressed in our experiment? Again, just a quick view of the SIRVs isoform module and what can be covered with the SIRVs. These mimic the full isoform complexity that we know today. They include alternative start sites, end sites, cassette exons, mutually exclusive exons, exon splitting and intron retention, and thereby they present a highly complex ground truth. And this can be used to benchmark and validate workflows and data analysis tools. It also means that this allows us to use the SIRVs as a known variable and to evaluate all our other results based on the outcome that we obtain for the SIRVs. If we can depict the SIRVs correctly, we can also be very confident that for most of our transcripts, we also have a correct solution. In this example, SIRVs have been used for exactly that. They were used to benchmark commonly used algorithms for transcript assemblies. And they were compared against Bookend, a newly described tool that incorporates end site information into the assembly. Single-cell data on mouse cells that was spiked in with the SIRV-Set was used to probe the accuracy of the assembly for the 69 isoforms. And as you can see in the plot, the truth is depicted on the left and the performance of the algorithms is depicted on the right. Bookend retrieved the majority of correct assemblies based on the known isoforms that are included in the SIRV spike-ins. To learn more about this, I would like to point you to our on-demand webinar with Michael Schon from Wageningen University. He's the first author of the study, and you can watch the webinar wherever, whenever. Just sign up with us and have a look at what the researchers did here. Another example I would like to show uses the SIRVs for the validation of differential expression workflows. This is something that is not so common. Normally, differential expression experiments work like this, that you are assessing samples between different conditions, so either treated or non-treated, or mutant versus wild type. And following the RNA extraction, as libraries are prepared in sequence, and afterwards what you want out of this experiment is a quantification of gene expression. How many more genes are expressed, which pathway do they belong to, and what is the response to the treatment. However, also here, how can we ensure that our experimental parameters are correct and our conclusions are valid? The number of differentially expressed genes, as you will see in the next slide, may not be as informative as widely thought. And it's not a valid parameter to assess the question at all. So the number of differentially expressed genes, as in the previous experiment, is much dependent on the experimental parameters and the data analysis routine you are choosing. And at this point, more does not mean it's better. To show you an example, here's the same data set that is analyzed with two different routines. And as you can see, the results are vastly different. While Tool A detects more than 2,300 differentially expressed transcripts, Tool B only delivers approximately 300. This goes in line with the observations that also the group from Wageningen had, where data analysis tools and settings can strongly affect the outcome of an experiment. So how do we know which one of these tools is now actually correct? And here I would like you to take you back one step and introduce the validation dataset that was used for this experiment. For all the samples, we used the same universal human reference RNA. And this RNA was then spiked in with SIRVs. So let's imagine the red circles on the image below represent the SIRVs. In the experiment, different samples, in replicates of course, were spiked in with different amounts of SIRVs, so that the concentration of the SIRVs is higher in one sample than in another. This also means that when we compare these sample groups against each other, we effectively generate a predefined log-fold change for only the SIRVs. In other words, while the SIRVs are the differentially expressed transcripts, in all our samples, the human transcripts of the reference RNA is not differentially expressed. So all human transcripts that are reported with a changed expression are actually false positives. And before we come back to the analysis, I would like to introduce yet a third tool for data analysis, MixSquare. MixSquare is a software that was designed for precise transcript concentration estimation. It does not assume coverage for transcripts, but it actually fits modules to the observed coverage, meaning that the algorithms can also learn to work with your data. This makes MixSquare highly precise in isoform quantification, and it allows extremely accurate detection of differential expression. MixSquare also offers a fast runtime and a small memory footprint. But now back to our experiment, and here MixSquare was also included as a third tool. And again, MixSquare quantifies the differentially expressed transcripts most accurately of all the tested tools. Because again, only the SIRVs, which are highlighted in green and the ERCCs in blue, are actually differentially expressed transcripts. Everything highlighted in red is false positive endogenous transcripts. So MixSquare detects only 37 of these false positives, while other tools detect over 300 or even over 2,000. In summary, the number of differentially expressed genes or transcripts, as we have seen, is strongly dependent on the chosen experimental settings. And as a take-home message, we can say that the sheer number does also not allow us to draw any conclusions about the suitability of the method or the library prep or the data analysis tool that is used in the experiment. Less is often more, and it means that you can potentially call true differentially expressed genes and transcripts with a higher confidence, and you won't get lost in the noise of the natural variations if you include the right controls in your data analysis settings. Therefore, spike-ins are essential controls for RNA-Seq experiments. Only they allow validation and monitoring of the RNA-Seq workflow and assessing the technical parameters such as biases, sensitivity, and length representation, and also the evaluation of functional aspects like isoform detection and quantification. This allows you to know the performance and the limits of your RNA-Seq workflow and data evaluation and draw your conclusions accordingly. Moreover, with spike-ins, you can evaluate comparably the samples in experiments and also between experiments. Therefore, you can evaluate technical variability between samples in experiments and before even starting to investigate the biological variants. Now that we have assessed the theoretical part of an RNA-Seq experiment, I would like to walk you through our RNA-Seq portfolio and point out where our solutions can actually offer benefits for the quality of your RNA-Seq experiments. Along the RNA-Seq wet lab workflow, we start with sampling. Lexogen provides several solutions for the sampling step, including SLAM-Seq for kinetic RNA sequencing, which allows time-resolved global assessment of effects and perturbation experiments. So if you are interested in RNA analysis and turnover, have a look at the SLAM-Seq technology that is exclusively offered by Lexogen. A second solution for sampling is our RNA-DNA Defender solution. RNA-DNA Defender is added to the tissue or cell sample prior to extraction, and it stabilizes the RNA inside the tissue at various temperatures. This allows to postpone RNA extraction, voids the need for liquid nitrogen to snap freeze your samples, and allows you to start your experiment with the best quality sample possible. The second step is RNA preparation. Here we offer several RNA extraction and isolation kits, including Trapper, a proprietary method to isolate functional small RNAs that are bound to Argonaute proteins. Trapper selectively enriches small RNA, and thereby it reduces the reads that are wasted on rRNA or tRNA degradation products. And moreover, it uses a gel-free method and only takes 15 minutes prior to the RNA extraction. Further, Lexogen provides solutions for poly-A selection and ribodepletion. And here we have also reached a decision point in any RNA experiment. How to choose between poly-A selection or rRNA depletion? The human transcriptome consists of roughly 85% ribosomal RNA and about 10% tRNA. The remaining percentage is then distributed between all the RNAs that are actually of interest to us, mRNAs, non-coding RNAs, and so on. So before deciding, you need to assess which RNA types you need to see. While poly-A selection will allow you to assess mostly coding transcripts, ribodepletion additionally offers a comprehensive view of long non-coding RNA, small nuclear RNA or nucleolar RNAs, circRNAs, and enhancer RNAs, which are often not polyadenylated, and they would be missed in an mRNA-Seq experiment. Lexogen not only offers solution for poly-A selection, but also for ribodepletion. And our RiboCop rRNA depletion kits enable specific and highly efficient non-enzymatic depletion of ribosomal RNAs and of globin mRNA. We use a non-enzymatic technology in order to preserve the RNA integrity of your sample. RiboCop can be used for a wide range of inputs, down to 1 nanogram of total RNA before the depletion step. So you can process very, very valuable samples with it as well. We provide specific versions for human mouse rat, kits for bacterial rRNA depletion, and for yeast, and there's more to come. RiboCop can also be used for work with whole blood samples, as we offer simultaneously rRNA and globin depletion. If you want to know more about how to choose between polyA selection and ribodepletion and which methods are out there, check our free RNA lexicon. This QR code will take you directly to the depletion chapter. You can also, again, get access to our free ebook and read the full RNA lexicon on your own time. At the beginning of this talk, we focused on how to choose the most suitable technology for library preparation based on your experimental needs. So for the NGS library prep part, I would like to come back to our technologies QuantSeq and Coral. QuantSeq is our flagship technology and has more than 1,000 publications already for gene expression profiling. It generates reads directly on the 3' end, and therefore it requires lower read depth and offers fast and easy data analysis through gene counting. QuantSeq libraries are prepared within 4.5 hours in only 5 steps, and you do not need depletion or enrichment upfront. The library prep is robust, and it can be used even for challenging samples, such as degraded RNA or FFP samples. QuantSeq kits include all components to complete the prep, including purification reagents and Lexogen's patented UDIs for high-level multiplexing with exceptional accuracy. Coral is Lexogen's fast and versatile whole transcriptome library prep. This also is an all-in-one kit that contains up to 384 UDIs for maximized sequencing output, all purification reactions, and it has built-in UMIs, unique molecular identifiers, for accurate expression quantification. The protocol is completed in 4.5 hours, and it is highly sensitive for low input and low quality RNA, including FFP samples. Last but not least, Lexogen provides data analysis solutions, including software. For example, Mixed-Square, as we have seen. We also have publicly available tools on GitHub to facilitate analysis of sequencing data generated with Lexogen library preps, and data analysis pipelines on our web-based platform. Stay tuned, because here more is to come. Thank you very much for your attention. Leave us a comment or reach out to us. We'll be happy to hear from you. Should you have any questions, please let us know at support@lexogen.com. For more information, follow us on LinkedIn, Twitter, or on YouTube to never miss a video. For now, have an excellent day and much success on your RNA-Seq journey.